Coronaviruses, you may notice a pattern here. We're going from smaller viruses to bigger and bigger ones in each different section. So we started out with the really small phage, got to the big phage. We start with the small positive strand RNA viruses. We're going to get to the big single strand, positive strand RNA viruses. The largest of these are the coronaviruses. They have the largest positive strand, actually the largest RNA virus genomes that we know of to date. So really pretty fascinating and kind of breaking some of the rules that we thought we knew about some of these viruses. Mostly coronaviruses were known for causing certain amount of the common cold, but of course what causes most common colds? The rhinoviruses, exactly. And then, again, basically, very few people cared about coronaviruses, although there was certainly some veterinary disease, and some coronaviruses and veterinary disease are a lot more of an issue. And until, of course, SARS came along, and then everybody cared about coronaviruses. So we'll talk a little bit more about that as we go along here. A couple of key concepts that we really haven't talked about, and again, sort of the idea here is that we're building literally and figuratively on kind of the things that we've talked about before. These viruses, most of them, but not all, have protease receptors, which seems at first glance to be kind of strange. Why bind to something on the outside of a cell which is going to chew up the virion? But if you think about metastable virions, it actually makes a lot of sense to bind to something which will then start to break down your virion. Whole idea of frame shifting. I don't really talk too much about frame shifting, but the frame shifting, which is where the ribosome shifts reading frame as it's translating, giving different proteins, turns out is used not only in the coronaviruses, but the retroviruses, a number of other viruses, which we won't be talking about. So this whole idea of having different amounts of protein and how do you get different amounts of protein, turns out that the frame shifting is one way that happens. But these coronaviruses and the related viruses are really fascinating because they have these things called subgenomic RNAs, which are really important for making proteins, and particularly those proteins which you need large amounts of. So the idea here is really that we need lots of our structural proteins versus our non-structural proteins, and so that's one way to do that. And another concept that Dr. Hirsch briefly touched on, but we really haven't talked about in too much more detail, is the whole idea of a reservoir species. And this is really important when we're thinking about human disease in particular, and a lot of the big, bad, nasty viral diseases, including things like SARS, and at the end, we'll talk a little bit about MERS as well. So, standard kind of outline, disease, where were these things originally found? A little bit about the structure, binding, and entry, but the big and interesting part, and new certainly as far as the viruses we've talked about so far, is this idea of subgenomic RNAs and making the RNAs from an RNA virus so that they're not completely full length. So we'll spend most of our time talking about that. Translation, the big difference there is this frame shifting, and then how these things are bound and released. So where do coronaviruses come from? Originally, again, they were studied looking at relatively benign diseases, all of these human coronaviruses causing pneumonia, and then a number of these veterinary diseases. Curiously enough, these are respiratory, whereas a lot of the veterinary ones are gastroenteritis. The canine coronavirus is fecal-oral transmitted, and so that's one of the reasons that it's one of the things that actually there's a pretty decent vaccine for. There's good vaccines for the canine coronaviruses, unfortunately for some of these other ones not. There are two different major groups. There's actually more than that now. Group 1 and Group 2. This is just how they're binding to different cell types. We'll see the difference later on. There are a few of Group 2s that are also causing these colds and pneumonia. There's even a hepatitis virus. This is a mouse hepatitis virus. So we've had hepatitis viruses so far that are picoRNA viruses, that are flaviviruses, and there's also some that are retroviruses, and yet alone a coronavirus. So livers are a place where you end up with lots of viral disease. And then, of course, the one that everyone has gotten really excited about coronaviruses for. And all the people who have been working on these kinds of coronaviruses beforehand were like, woohoo, we're actually going to get some funding, so they could continue to study their particular coronaviruses. So this is, of course, what happened at that point. Some of us hopefully still remember this. It was a few years back now, about 15 years, but SARS was going to kill us all, and it was spread all over the news and newspapers, etc. So what do people know about coronaviral diseases before SARS? It was pretty minimal. Common colds, nobody really cared that much about them. Again, a lot of it had to do with these veterinary diseases, and people were studying that, and then they were just these weird kind of diseases that were made by these viruses that didn't look like other viruses and had these really big genomes, which was interesting from a scientific point of view, but not really so much medically. And a lot of the work, and in fact most of the stuff that's written up in this chapter, really has to do with murine hepatitis virus, because that was a relatively easy virus to work with in the lab. So a lot of the stuff in terms of what's in the textbook is really about this. But then SARS came along, and I actually kind of think of this kind of an epidemiological triumph, really. Severe acute respiratory syndrome, first reported in February of 2003, and they figured out that it was a virus a couple of months later, and then after that had the genome sequence, and were really able to control the disease quite rapidly. There are a couple of caveats that we'll get back to a little bit later on. But from February to March, there were over 1,000 cases, so it really blew up in sort of the typical part of the word. So what is SARS? Severe acute respiratory syndrome. Basically starts out with feeling crummy as if you have a cold, but progresses really rapidly. That's the whole acute respiratory syndrome. And what happens is you end up actually, your lungs completely fill up with liquid and you can't get enough oxygen. That's what happens in these diseases. There were overall almost 8,100 cases and about 700 deaths. So about 10% of the people who were diagnosed with SARS ended up actually not surviving, although it got much better over time. In the beginning it was worse, and then it got much better. Given all of that media attention, there are only 8 cases and 0 deaths in the U.S., partly because it actually got to the U.S. a little later than places like Canada. So about 10% of people, again, in these cases, relative to the 50+% that we'll talk about with Ebola, and actually even later on with MERS, as when we get there. How do you get SARS? This is actually probably one of the reasons that it didn't spread that quickly. It does seem to be direct contact and aerosols. So somebody is sneezing in your vicinity, then that's how you can end up getting SARS. And that's why you told the people with the masks, that seemed to help actually quite a lot. But also it was getting care and diagnosing the disease very rapidly. So as soon as the disease is diagnosed, then you can get the appropriate treatment. So that made a big difference. The question, of course, is where did it come from? It seemed to come out of nowhere. The original cases that they found were in people who were associated with some of the live animal markets in China. And the first thought was it was these civet cats. These were the civet cats, which I'm sure many mammalogists here. I think they're neither related to cats nor much of anything else, but are quite present in a lot of these live animal markets. The genome of the coronaviruses found in civet cats was actually practically identical to the one that was found in humans. And so everyone was like, oh, it's these cats. We get rid of the cats in the markets. We won't have any problems. But then people started to look a little bit further, and they found that there are a lot of bat coronaviruses. And in fact, we knew nothing about bat coronaviruses until SARS came along. Now we know a lot about bat coronaviruses, and particularly the horseshoe bat. And then we'll talk a little bit about this as a potential reservoir species a little bit later on. So 8,000 cases, 700 deaths. over 4,000 citations. So for every case, there are like 2 papers that were published on SARS, which I think is quite impressive. But just to give you an idea what the activity was, in 2002, there were 120 papers on coronaviruses that were published, and in 2003, there were 620. So just a huge increase, and now to 4,000 papers. There were 4,300 in last year, and then 4,410 this year. So even like 100 a year are still being published on SARS, even though there haven't been any cases of SARS since 2004. So it came up and was gone really, really quickly. Although even though there haven't been any SARS cases, SARS was declared what's called a select agent. Now, we haven't really talked, we'll talk more about this when we talk about some of the nasty diseases like smallpox, etc. There are multiple biosafety levels and risk levels, risk groups for different pathogens. So risk group 1 are the things that you could basically drink and you'd be perfectly happy with. Risk group 2 are things that you have to be a little bit careful about, including actually most of these flaviviruses. Risk group 3 are those that can cause nasty diseases. Risk group 4 are the ones that cause really, really nasty diseases. And the select agents are a small group of that risk group 4 organisms. And the select agents are those that if you want to work on them, basically everyone in your lab, everyone that you work with, everyone in your institution, everybody you know, probably that's a little extreme, has to do background checks and everything else. So working with any of these things is extremely difficult. I think it's a little strange that we haven't had cases since 2004 and then 9 years later they decided it's a select agent, but I'm not the person who makes these kinds of decisions. So let's revisit this whole idea of a reservoir species. And so this is a very important concept that, again, Dr. Hirsch covered a little bit before, and this will come up again and again, particularly when we talk about some of the negative strand RNA viruses a little bit later on. So the idea of a reservoir species is sort of goes back to our whole thought about viruses and definitions of viruses in the first place. You know, viruses are dependent on a host, and in order to be able to replicate and be maintained in a population, you have to have this kind of hosts producing virions, which infect a host, and this whole idea of a cycle. If you kill off most of your hosts, it's not a really good way to be able to persist as a virus. And so, Lambda figured this out and just hangs out as a lysogen. And as a Lambda lysogen, E. coli are perfectly happy. And so this is sort of the analogy to what's happening in a lysogen. Many viruses seem to just hang out in a host, don't cause them any kind of obvious disease, but are still replicating. And then in some cases, those then viruses will make a jump, as people call it, from one species to the next. And very often in that second species cause some pretty nasty disease. And that's exactly what seems to be happening with SARS, almost necessarily is happening with Marburg, with Ebola, and potentially a lot of the things that are causing nasty diseases in humans are being maintained in some non-human animal, and in those cases they seem to be relatively fine. And a lot of people are now finally, in my biased opinion, starting to look at some of these native potential reservoirs, and then why are they not causing disease in some of these animals, and why are they causing disease in humans? What's the difference between some of these things? And in the case of SARS, it does appear that the reservoir species for SARS is in some of these bats. And part of the reasoning for that is that if you just look in the Philippines, 50% of the bats have coronavirus RDRP. And when you say in bats, mostly what that is going through bat guano and amplifying whatever RNA you can find with reverse transcriptase PCR, and you see RDRP sequences. And you can then very closely align those to different coronaviruses. And as we'll see in just a second, turns out that there's a huge amount of diversity in coronaviruses in bats. don't seem to have any symptoms whatsoever. And it's not just in the Philippines. Another study was done, and these are just, by the way, are references if you want to look these up. In the US, about 20% of the samples that they tested were positive. And I have to be careful about this one because it was a relatively small sample that they looked at. So 20% of, I think, 30-odd bat samples. So it's distinctly possible that these numbers are pretty variable. But certainly there are bats that have coronaviruses that are associated with them, and again, seem to cause no disease whatsoever. So when you get all of these sequences, what do you do with them? If you're good molecular biologists, you do phylogeny, where you take all the sequences, you line them up next to each other, you feed them into a computer program, and it spits you out this tree. So how do you try and read some of these things? Basically, the length of each of these lines represents differences between, say, these sequences here and a presumed sequence that you had back up here. So if you have practically no line here, like, for instance, you have in this case, SARS in human and SARS in civet cats, these guys are practically identical sequences relative to each other. So that's one way of looking at these. But then what you do is you look at groupings. And so all of these sequences here are grouped together and in fact, the best way of looking at it is right here. All of these sequences grouped together. And this 100 right here means that if you do this analysis, 100 times, you end up with all 100 having this branching pattern right here. On the other hand, if you do this analysis 100 times, 23% of the time, all of these guys will group together. And so anytime you see one of these bootstrap numbers or confidence numbers at any one of these axes, you immediately think to yourself, okay, if it's, under 50% or under 70% of the time, I wouldn't necessarily believe that. If it's above 70%, then that's usually figured as being a pretty good estimation of what the relationships are of these. And these are also supposed to represent a history of these different sequences. And so if you go back to here, this would be the common ancestor of all of these other different kinds of viruses and so on and so forth. And so the take-home message here, really, at least as far as SARS is concerned, so SARS sequence was determined very quickly, found to be identical to the SARS sequence in these civet cats, but these were, not seeming to cause disease, and very few of the cats actually had any of these coronaviruses. On the other hand, all of these other sequences here are bat viruses, so it certainly looks as if there's a lot of diversity in these different coronaviruses, and there was this bat virus, again, the horseshoe bat virus, that's very closely related to these, not very much sequence difference, and it branches together with these guys all of the time. So highly likely that it was this bat virus that either went directly to the humans or ended up at the cats, and those end up at the humans. Yeah?
So when you're attenuating a virus to give as a vaccine, you're running it through like bovine or chick embryo cells, and you're weakening it.
Mm-hmm.
How is that? It seems like the opposite thing is happening in nature when it's running through a species of like bats, and then it jumps to a human and it's suddenly super terrible, causes disease. What is the difference? What's happening there?
So I'm going to paraphrase your question, if I may, and correct me if I'm wrong, Anka. So basically, all this attenuation happens, and shouldn't it also be being attenuated by circulating in one of these reservoir species? That's sort of another way of thinking about it. So the process of attenuation, what you do is you take the virus from one particular individual and then reinfect with that exact virus some other individual. And usually this is actually causing disease in those cases. And what happens is you're kind of doing the selection for, in that particular organism, causing less and less disease. You start out causing disease and then less and less disease. So presumably in that evolutionary process, the disease is going down. Now, the next thing you have to do with that, however, is then test it in other organisms, like humans or non-human primates, and see if you end up with something which is attenuated. What happens is when you try and do attenuation processes, at least this is what I've read, is that sometimes they're not attenuated at all. And those are the ones you throw out.
Okay, so this is manually selecting, like,
You are actually...
This one looks like a scary, let's get rid of it.
Exactly. And so that's my understanding, is that what's happening there. And in this case, very often for these attenuations, I don't know if you remembered from when Hirsch was talking about the attenuation for yellow fever, multiple different kinds of organisms. So they had chicks, they had, I think, mice as well, and brains, and so lots of different things that they were passaging them through. Whereas, so they don't have that opportunity for the host then to evolve and the viruses to be co-evolving with them. And so the idea there is that the viruses are evolving faster than whatever virus-host interaction would be. And so those are sort of, it's hand-waving, to be perfectly honest. But that's sort of the idea, is that they're probably attenuated, and actually really clearly attenuated in bats, but not when they then get into humans. And again, this is why I think these are people fortunately now are looking at this, is why do you have bats be, and there's actually a really fun TWIV about this, and if you listen to This Week in Virology, talking about differences in bats and bat immune systems versus what we have in humans and human immune systems. Okay, so we're all happy about reservoirs and that kind of thing. This should now be really easy. Reservoir species 4, SARS coronavirus, is probably humans, bats, civet cats, rats, or mice. Let's start. Oh, you can't see this, sorry. Let's move this across. Around. Everybody voted appropriately? I think I have everybody's clicker numbers now, so I should be able to upload them all. And give you all one guess what it was. Oh, come on, let's show the results. There we are. Yes, it's the bats, as far as we can tell. And actually, again, really fascinating question about bat and bat immunology. Now, if you're trying to work with bats that are infected with these diseases that can cause 10% or higher percent mortality in humans, actually having a facility where you grow bats and have them controlled in a lab is not straightforward at all. And so there are only a couple places in the world that are actually doing this. Yeah, Christian.
So can we go back to the tree again?
Yeah, sure. Oh, sorry, that's one too many. And then I can hide this. Yeah.
So I find it weird that the range length of civet cats is about the same as the human range.
Yeah.
So that makes me again, do we have like a correlation with the range length and how few preventive virus would be in like even species?
So I'll paraphrase your question if I may. Basically, do the civet cats get sick? And is there any difference? Because the sequence here are basically identical to each other. And is it a sequence thing which is causing a difference in terms of disease, or is it a host thing? Is it a virus sequence, or is it a host? I think is the question. And if I remember correctly, and this was a while ago that I went back and looked at it, I think the cats were pretty sick too. But that's a guess. But what was found, and again, I'd have to go back and look at this, were that the cats that were in the markets were infected, but those that weren't in the markets weren't. And so it seemed to have been something that was really proximal to getting this transmission happening. But again, I don't remember all the details on that. I can send you all the references. You can check them out. But yeah, the sequence there, and even the sequence here, this distance, to get back and look at this this table here, this bat SARS-CoV sequence is still very similar to these sequences, and these bats seem to be perfectly fine. So at least in the case of the bats, there doesn't seem to be a really big issue there. Yeah. So
if you step back one to the common ancestor,
yeah.
let's say that 37,
Mhm.
37
Yeah.
times that they ran it, they got,
Right.
and so that's just the highest that they got.
Yeah, well, that's they got 37 where these branch together, and the way that these training programs work. This is a great point. It's what I prefer to do when I look at trees like this. If I were the reviewer of this paper, I would say put a dot there, because 37 means that 63% of the time they didn't branch together, but they're still branching together with some of these subsets. And so, yeah, no, I do not like this tree particularly, certainly for these deeper branches. These ones are pretty good.
Right, yeah, I can see why if it was getting to the 90s and the 100s,
Yeah.
it just seems like if you step back one that clearly something's missing.
Yeah.
In there that it's not allowing to.
So probably what's going on here, and this is just thinking about my knowledge of phylogeny, which is limited at best, is that it means that there's actually way more sequences that are there that we just don't know about. And very often, if you add more sequences to these phylogenies, those things start to tighten up and you get better bootstraps. No, and I forget, you could go and look in NCBI, the number of coronavirus sequences. It's probably in the numbers of thousands now. And the other issue is when you try to do some of these phylogenies, the larger and larger they get, the longer and longer it takes to do these kinds of analyses. And so we've been doing some analyses in my lab of about 500 sequences that we'll hear about a little bit later on. And those take a week to run. So, yeah, these take a little while. Okay, other questions on this stuff till we finally start to talk about the structure of these things and how they replicate themselves? Okay, so if you just look at these virions, they don't look very nice and similar to each other. And that's why I don't have a nice model of coronaviruses, because they're pretty pleomorphic. They're very different in terms of their structures, and the virions are not all the same at all. But what they do have are these spikes around the outside, and that's the crown. This is why the coronaviruses, so corona for crown. Enveloped viruses, inside the envelope, most of them have helical nucleocapsids. So instead of our nice regular icosahedra, these seem to be packaged as helical nucleocapsids. And we'll see this is true for a lot of the other enveloped viruses a little bit later on. Quite why that is for the coronaviruses is not entirely clear. It is much clearer for some of the other ones. A few of them have icosahedral capsids. So what are these made up of? They've got the crowns. These are the spike protein. And so this spike protein, this should sound very familiar, is a trimeric protein. It also has the fusion peptides, where it's going to give you the fusion of the membrane of the coronavirus with a host membrane. Turns out this happens in a couple of different places. Some, but not all coronaviruses, and when I say some viruses and the ones that people care about, turns out that SARS is one of those some, has a hemagglutinin esterase, also membrane protein, that has hemagglutination activity, a lot like flu. We talked about the hemagglutinin beforehand. And neuraminidase activity, which chops off sialic acid, so it's helpful for release of the virus particle when it gets inside the cell. So again, some of these, and SARS in particular, has this HE protein. The most common of these proteins, interestingly enough, is this protein called M, which is called membrane protein here, but is usually called matrix protein in most other envelope viruses. And so this is what provides the bridge between the nucleocapsid on the inside and all of the spike proteins, the envelope proteins. And getting back to your question, David, about when you have the host proteins sitting in the membranes, it really does seem to be these matrix proteins which are holding everything together, and then the nucleocapsid comes and forms right there. And so it's kind of the glue or the adapter that you have between those membrane proteins and the nucleocapsid proteins. The nucleocapsid is not going to go anywhere in the membranes. It's going to just go where those matrices are. And so that actually turns out is the most common protein. So if you just purify coronavirus virions, that membrane protein or matrix protein is the most abundant of them. And people have shown really nicely it interacts with both nucleocapsid and spike protein. And nucleocapsid is just this nucleic acid binding protein against binding to this single-stranded RNA. And then in some cases, you have this envelope protein that may be involved in budding, but you can get rid of it if you don't actually need it. So not surprisingly, it's the spike proteins that are going to be interacting with receptors, and I mentioned this right at the beginning. These interact now, in most cases, with peptidases on the outside of the cell. And again, this is not surprising at all, because if you have a stable virion on the outside of the cell, one way to make it unstable is it binds to a protease, and that protease will break down part of the virion so that the genome can be released. So it makes a lot of sense. The actual names of these things are not important. That they are proteases, I think, is quite important, just again because it's one way that you can get inside the cell. And one of the ways that group 1 and group 2 are different from each other, there's that sequence, and actually I kind of mentioned that before. If you looked at phylogeny, one of the things that actually did have 100% bootstrap was the group 1 versus group 2 viruses, but also they interact with different receptors. Those that have the HE protein in them are often also going to interact with sialic acid. And so hemagglutination, getting the red blood cells to bind to each other, is almost always sialic acid. Sialic acid is present on particularly lung epithelia, and so that's how a lot of these things seem to be happening and binding to these respiratory viruses getting inside the lungs. So one of the neat things about these spike proteins, and also partly because people were really excited about SARS, is that high-resolution structures have been determined for many of these, and this is actually, this is a paper by David Wiesler and his group from University of Washington. They solved this structure of the trimeric S protein, and actually just the part which is outside the membrane here, with these nice fusion peptides at the top here. Looking down at the top, the fusion peptides are sticking out. They only get stuck out after you have proteolysis that takes place, and so that's how you end up getting the membrane fusion taking place with that. And actually, David was here a couple of years ago. Anybody hear his talk? I think it was probably before the time that most of you were here. He talked a lot about this structure when he was here. So you have the binding, releases, membrane fusion. Sometimes it's the plasma membrane, but also in endosomes. It depends from virus to virus. Then you get these ginormous RNAs, which get released inside the cytoplasm of the host. And these are the largest positive strand, single-stranded RNA viruses, and actually the largest RNA virus genomes known. The only one, I think the one that actually has a virus associated with it is, I should say virion, is 31,000 bases, but recently people have been doing metatranscriptomics. Anyone know what metatranscriptomics is? Sequencing all the RNA in an organism. So when they did this, they thought they were going to get all the messenger RNAs, and they actually ended up with a bunch of virus sequences. And so they were looking in planaria, which are flatworms, and aplysia, which is a mollusk, and they were finding these really humongous genomes, 41,000 bases in the flatworm and 35,000 bases in the mollusk. And basically what it means is there are lots of these things in all kinds of other places that we've never studied them. Who cares about viruses of flatworms? But could tell us something about how these viruses work. The real surprise with these things being so big is that RNA-dependent RNA polymerases don't have proofreading activity. And they're actually not very high fidelity. So people had actually made predictions that about 10,000 nucleotides was just about as big as you could make one of these RNA genomes and still have it be replicated, and not every generation having so many mutations that they would no longer be functional. So the fact that these things actually do have these really big genomes have had people then revisit this whole idea that RNA-dependent RNA polymerases and reverse transcriptases, when we talk about retroviruses later, have very high mutation rates. So they do seem to have very high mutation rates, but there also seems to be some corrective activity, which is happening with them. And quite how that works, I think, is kind of up in the air. So, was RNA-dependent RNA polymerases, RNA virus genomes, why don't we have all of our genomes be RNA? Because in an RNA world, that was, probably was. RNA was the genetic material and was the catalytic material. Why did we switch to DNA? Well, DNA works because it's more stable and you can, have DNA polymerases that proofread, but we have RNA polymerases that proofread as well. So why not? So that's just fascinating kind of an aside thing. But if you look at the genome, now you look at the translation products, the potential translation products of the genome. First is these have caps and tails. So regular poly tails, regular caps, and big polyproteins, but not the whole thing. And so it turns out that you've got one set of polyproteins here, known as ORF1A, and another set of polyproteins right here, ORF1B, but they're in different reading frames. And just slightly offset from each other, and we'll see in just a second, it's all about the frame shifting process. But then all the structural proteins are down here at the 3' end. And they're all in separate open reading frames. So something weird is going on here in terms of the translation. And we'll get back to that and talk about it in a little more detail. So a couple of proteins. Unfortunately, the names here are ridiculous. I don't expect you to remember them. But some of the activities are really important. First one is, not surprising, it's a polyprotein. What happens with polyproteins? They get snipped up. What snips them up? Proteases. So there are multiple proteases, particularly these non-structural proteins, NSPs. When we look at a lot of these things, that NS is going to be non-structural, or NSP is non-structural. We already talked about the hemagglutinin spike envelope membrane and nucleocapsid proteins here. And then, just like you have in all of these other RNA viruses, you have an RNA-dependent RNA polymerase. So RNA-dependent RNA polymerase, probably this NSP12. And then, remember, we've got caps and tail structures. How do you get caps and tails, particularly cap structures, if you're replicating in the cytoplasm? Where do those come from? They're viral proteins, so viral methyltransferase proteins. These are what are involved in making caps. And so I forgot to talk about this the first time I talked about plant viruses, but then I corrected myself to go back and talk about the methyltransferases. So many of these things have RNA-modifying enzymes, methyltransferases, etc., exonucleases, etc., which are involved in modifying the RNA. Because all of this is happening in the cytoplasm. You don't have access to all of the other RNA modification enzymes that you'd otherwise have in the nucleus. So all the RNA modification is happening here. So getting these non-structural proteins, they're, as I mentioned before, these guys are out of frame relative to each other. And what happens, there's a secondary structure in the RNA right at this junction between the two. And what happens is the ribosome basically gets a little confused and will step back a nucleotide and then continue to translate in the different reading frame. This doesn't happen all of the time. And what it means is you have less of these proteins here at the end than you do have of these proteins at the beginning. And that kind of tells you, as far as the non-structural proteins are concerned, that these proteins that are encoded down here, you need more of these than you need of these here. And particularly important are these proteases. So proteases, proteases, proteases. These things you need immediately. Again, not surprisingly, you're translating your big polyprotein, and those then need to be made to chop up the polyprotein into its smaller pieces. Once you've made these non-structural proteins, then you need to start to make genome. How are these genomes made? They're made on cytoplasmic vesicles, again, probably mostly because it needs to protect that double-stranded RNA from getting degraded. How do you get these vesicles? It appears that in a number of coronaviruses, what's happening is you have the autophagy process, which is basically cells eating themselves, and that eating themselves process ends up generating lots of internal vesicles inside the cell, and the virus just seems to take advantage of that, use these for replication purposes. As true for almost all of these positive strand RNA viruses, you make negative strand, but much, much more of your positive strand. And this is probably, again, just the same kind of thing as we saw way back when we talked about those RNA phage, is that when you're translating and replicating, they're going in opposite directions. And so getting that negative strand is a lot harder, but as soon as you got the negative strand, you're not translating in that direction anymore, so you can make a lot more of your positive strand. Don't ask me what the primer is for replication, because I don't know. But I did want to really briefly, right at the end here, talk about how we're getting our structural proteins. So, as I mentioned before, these are all separate open reading frames. This is no longer a polyprotein. Down here at the end, how are these being made? And the first hint in terms of how these were being made is that all of these are actually, when you purify RNA from infected cells, you have a bunch of these shorter RNAs, each of which has a cap on them. But they also have the same sequence that is present at the 5' end of the genome. So what's happening in terms of making these RNAs? And this, again, these are your structural proteins. So this is what you need the most of. How does that happen? It turns out that the RNA-dependent RNA polymerase starts out making negative strands at the end of the genome and copies until it gets to one of these so-called translational regulation sequences, or TRSs. And occasionally what will happen, at a certain frequency, what will happen is then the polymerase will fall off and then go over to the 5' end and copy this last little piece here. And this happens, again, at certain frequency, but what it means is you've got lots and lots of these really small ones, and some longer ones, some longer ones, some longer ones. And those are what ends up getting translated and giving you much more of your structural proteins than of your full-length genome or of all of those non-structural proteins. So this idea of what's also called nested subgenomic RNAs, and we'll talk more about this with some of the other viruses, which actually do exactly the same thing. These are getting you more RNAs for your structural proteins, more RNAs, you're going to get more translation, you're going to end up with more of those structural proteins. Once you have structural proteins, then you can assemble. Assembly here seems to happen actually very similarly to the flaviviruses that Dr. Hirsch was talking about. You have formation of your membrane proteins together with the matrix, and then nucleocapsid. This buds into the Golgi. Regular excretion process will end up getting this to the plasma membrane, and those are then released. I think I'll actually stop here and not talk about our recombinant coronaviruses, and we'll do a little MERS intro on. Wanted to briefly finish up with coronaviruses because we didn't get to the fun parts last time because, well, the most fun parts last time anyway. We ran out of time because of all of my technical issues. So today I did want to very briefly just talk about what I think is a much more concerning aspect about coronaviruses and coronavirus disease before we shift over and start to talk about measles and why vaccines are such a wonderful thing. So last time we talked a little bit about SARS, severe acute respiratory syndrome. That came up 2003 and then was rather rapidly gone, probably about 8,000 cases, about 800 deaths, about 10% mortality. People got really up in arms about this. Very few people, however, know about MERS. MERS, Middle East Respiratory Syndrome, is much more of an issue, but I guess because there haven't been as many cases here, it hasn't gotten anywhere near as much press. So MERS coronavirus, the first indications of this were in 2012, Middle East, and then also some cases in the United Kingdom. 30 to 40% death rate, so 3 to 4 times higher than for SARS. How many of us heard about it? Was it all over the newspapers everywhere? No. However, if you were in Saudi Arabia, it was, because that's really been the major center of this. Index case, we haven't really talked too much about index cases before. This is more of an epidemiological term or an outbreak term. The first case of where someone was diagnosed with this disease was in September of 2012. And then it's been going on for a number of years, again, mostly in the Middle East, partly why the Middle East Respiratory Syndrome. And as of, this is all totals now, about 2,400 cases, 800 of those are fatal. So still very high rate of mortality for this particular coronavirus, probably partly because of the fact that we figured out that SARS was a coronavirus. People were looking for coronaviruses, and this particular one was also isolated in Rotterdam. And in fact, quite soon after the discovery of the disease, complete sequence, so between the first case and the complete genome sequence was about 6 months, which also tells us that we're doing a pretty good job even with some of these emerging diseases. And it became really obvious that this was a coronavirus, also, strangely enough, closely related to bat coronaviruses. And again, this should be no surprise based on what we talked about last time about the huge diversity of coronaviruses in bats. And so, even though, as we'll see in just a second here, people are also talking a lot about camels, which we'll get to a little bit more detail in just a second, is probably a reservoir in bats, and then is transferred via camels to humans, and potentially also that was what was going on with the civet cats in the markets in terms of SARS and the emergence of SARS as well. Just as a quick reminder, this is a classic coronavirus, also interacts with a peptidase receptor. So these peptidase receptors, let's see, who are we at now? Who am I going to put up? Christian, do you remember why these peptidase receptors are potentially so useful for coronaviruses? Here, I'll throw you the virus, it'll help. You want to throw it to someone else? Who else would like to answer why peptidase receptors are so useful for coronaviruses? Diana? To some extent. They're not that common on different cell surfaces. Any other ideas? Anyone else want the virus? What about virions? What's one of the really fascinating things about virions? They've got this metastable state, right? Could be stable on the outside, unstable on the inside. Echo has an idea. Throw it the virus. Yeah, so the idea, now this is not completely proven, but the idea is that if you have a peptidase that you bind to get inside a cell, that will also help to break down the virion. And so that's a way of helping genome entry. And so that's one of the reasons that people think that this makes a lot of sense. So that being said, again, classic coronaviruses, mostly in the Middle East. And interestingly, this is just some data, they should say these are some data from the WHO, looking at various outbreaks. The color coding here, dark blue is Saudi Arabia. So you can see that's really the vast majority of cases are in Saudi Arabia. But there's this one blip right here in 2015, the Republic of Korea. And this is South Korea, where they had a single person who came into South Korea in May of 2015, ended up with 186 cases, most of them in hospitals, and 36 of those people died. And as hopefully most of you know, this is one of the main medical centers in Seoul. And so it was really kind of frightening that these are such high fatality rates with some of these diseases. So fortunately, they were able to, mostly through quarantine, actually. It turns out that the way that MERS is spread is through very close contact. And as long as that close contact is avoided, unfortunately, medical care usually depends on close contact, that that was taken care of, and just a few months later, that was gone. Why haven't we heard about it? Probably because there haven't been very many cases in the U.S. Two, both healthcare workers from Saudi Arabia who got really sick really quickly took themselves to the hospital because in Saudi Arabia they knew about this, were immediately tested for MERS, were found to be positive, and there's no good therapies for these. Just had them stay in the hospital and be covered for a while, and then they were fine. But given those two cases, there were almost 1,000 people who were tested for MERS and found then to be negative. So that's where the state is, and again, probably why not all these people have heard about this coronaviral disease that has a 20 to 30% death rate. Where does, I should say, MERS come from? I mentioned Saudi Arabia. Because of it really a focus in Saudi Arabia, people have thought about, okay, where could it be coming from? And they found very quickly that people who were coming down with MERS very often had close contact with camels. And I do not know the culture in Saudi Arabia, but apparently a lot of the camel workers, people have camels as pets. They also have racing camels and are in very close proximity to the people they are working with. And they've gone back, they've looked at these camels for over 20 years, so well before the first, at least diagnosed cases of MERS. Clearly, MERS has been in camels for that period of time. There are some vaccines under development, actually mostly for camels rather than for humans. The numbers here, just a couple of thousand cases, so it's still a relatively low number of cases, but a certain pretty high proportion of the camels do seem to be actually exposed to disease. And it has an almost identical sequence to the human sequences, and so it's probably pretty clearly the connection. The camels seem to get a little bit sick, but not completely. What's a little strange is that people have found that there's MERS in camels in a lot of other parts of the world, but don't seem to be being spread to humans. And so a lot of this actually may be social. Apparently in Saudi Arabia, people get more up close and personal with their camels than they do in other parts of the world. So again, just a guess here. And there's a nice TWIV that talks about this in quite a lot of detail in terms of MERS in camels. So just as an overview, this is where the vast majority of cases are, again, in Saudi Arabia. Some of them imported. Those are those two that came into the United States here, originally found in the UK. And it is possible, actually I should have mentioned this, to get human-to-human transmission of the disease, but only, again, if you're in really close contacts. And it basically seems to be there'll be a couple of close contacts that get the disease, and then probably there are actually other people who are exposed and actually aren't coming down with the disease. They just never get infected enough to be detectable. But where we have the main transmission is really going on here in Saudi Arabia. So probably what's happening goes from camel to camel. In Saudi Arabia, it'll go to humans, and then occasionally those humans will spread the disease elsewhere, which is another reason why vaccinating the camels is probably the best way to get rid of this particular disease. So that was all that I wanted to talk about as far as coronaviruses are concerned. Any more questions on the coronaviruses? Yeah. Yeah, so the question is, what basically, what's the yellow? And so the yellow is where they've done testing of camels, and you do see camels with MERS in those locations. Now, what I don't know is where the bat coronavirus that corresponds to this circulates. That's a more open question that I do not know the answer to. Okay, so everybody's happy with coronaviruses?